I'd like to welcome all of you who are coming to Sol's lecture and didn't attend Bob's lecture as the chair of the Salk Medal Committee, and just tell you a couple of words about the Salk medals. They were inaugurated in 2005 to celebrate the 40th anniversary of the Institute's opening of its lab facilities. And the medals were established to recognize outstanding people in basic research and in public service, leading to improvement of human health. And in the 2005, the two medals were awarded for excellence in research to Don Metcalf for his pioneering work on discovering growth factors that regulate blood cells, which have subsequently been being used in the clinic, and Paul Farmer for his humanitarian work in setting up affordable or accessible healthcare to rural communities in Haiti. Then in 2010, so we had a 5-year lag, the medals were awarded to celebrate the 50th anniversary of the Salk's letter of intent, which was to found the Salk, which was in 1960. And the medals were awarded to Bob Rader for excellence in research for his discoveries, pioneering discoveries of how genes are transcribed into RNA. And the Public Service Medal was awarded to our very own Owen Jacobs for his philanthropy, particularly in supporting in basic research on the Mesa. So now fast forward 5 years again, and here we are celebrating no particular anniversary, but we are, as you know, awarding medals to Bob Weinberg for excellence in research, and that is shared with Sol Snyder. And so I'll simply ask, I should say one more thing. The Medal for Public Service was previously awarded to Itzhak Perlman in 2014 to recognize his, both his wonderful music, but also his efforts in eradicating polio through public speaking. He is, as you know, a polio survivor. So now I'd like to ask Rusty Gage to come and introduce our second medal lecturer, Sol Snyder.
Okay, thanks everybody for coming. I'm particularly happy to be able to give this introduction. I met Sol in 1972 when I was a graduate student at Johns Hopkins University, and Sol had arrived in '66, I believe, and was already sort of a paragon for those of us who were budding neuroscientists before the foundation of the Society for Neurosciences even. But we were all sort of thinking of ourselves as neuroscientists, and Sol was leading that charge. I had the opportunity to take some classes from him and mix with his students, and it was, and I should say over the years that Sol has been always very supportive of me throughout my career, and I've learned a lot from him by watching his behavior and how he interacts with people. It's been great. A little bit about Sol. He staying in one place, Ender was making this comment about Bob. Sol was born and raised in the Washington, Baltimore area and did his schooling there, and then did his undergraduate at Georgetown University, and then medical school and resident training at Hopkins. And probably as formative as anything, spent time with Julie Axelrod at NIMH and did a lot of his training there, and I'm sure was inspired, as many of us were, by Julie. I was thinking about some of the things that Sol has discovered over the years, and a lot of the things that we take for granted now as just being, didn't we always know that? Were actually at one point unknown. And he pioneered this reverse ligand binding assay that allowed him to discover the opiate receptors that exist in the brain. And this was such a, I mean, you have to live through this period where you realize that the opiates that are involved in addiction and related molecules actually exist within the brain. So there's endogenous opiates, and they have these very specific receptors that he revealed to us. And it changed, I think, this concept of reward, concepts of lots of things by showing that they exist endogenously, and then people could begin to manipulate them. But using the same technology, he did the same thing for almost all the neurotransmitter receptors, which was then incorporated by most of the pharmaceutical companies at the time to develop a lot of the pharmaceutical drugs, psychoactive drugs that we are currently using, are as a direct result of the pioneering work that he did. So that, that could be a career, and it has been, and plus the career of many other people as well. But then he went on to show that gases can be neurotransmitters. I mean, he actually showed that nitric acid, and this was another one of these hard things to really think about, that a gas as a neurotransmitter, and he showed that this could regulate glutamate. And he synthesized the nitric oxide synthetase, and then went on later with monoxide to show that that was also a neurotransmitter. I can really go on for a bit longer. I'll just say two more things, just so this whole class of research then that expanded on the gases and still goes on. I think you may be talking about it a little bit that today. But he also went inside the cell to look at inositol triphosphate and discovered the receptors for that, and which led to the discovery of immunophilins, the neurotrophic factor and neuroprotective effects of immunophilins, which is a whole other area of research that has been very prolific. I'm not going to go into this, but he also cloned many of the receptor binding proteins for the olfactory odorants that we all smell and use, which has also been adopted by industry for a variety of different reasons. There are other things, but there's a variety of books that have been written about him and stories that have been written about his life, and I encourage you to read them. For all this, he has received many prizes, probably, that it probably deserves many more. But among them are the Lasker Award that he was awarded, as well as the Wolf Prize that Bob was awarded, and is a member of all the major academies. He's written several really terrific books, too, that are worth reading, including Madness of the Brain, The Troubled Mind, and Brainstorming. So these are approachable books for the public, giving insights into psychiatric training. So for all this, we're honoring Saul, but there's one little bit more, and I would say that Saul is probably one of the most sincere and kindest people that I've known. He has an army of students and postdocs, you can imagine, that have trained with him, and they admire him dramatically or very emphatically, but more importantly, like him. He's just a very nice man. And on top of that, they named the Department of Neurosciences after him, so it's the Saul Snyder Department of Neurosciences. So it's my pleasure to introduce to you today Saul Snyder, Salk medalist.
Thank you very much, Rusty, who's been a dear friend for roughly 40 years, as he indicated. Let me just check this. Okay. I'll try to talk today. When you get to be a grandfather, you can spread your wings widely and talk about anything. As you can see, my granddaughter Emily lets everything hang loose. But at the same time, you can see she and her sister Abigail have vision, and this is a vision of a beautiful pond in Cape Cod where we would spend summers together. Time goes by, and Abigail is now a freshman at Kenyon College, and Emily is in the 11th grade looking at colleges. And people like that make life worth living. They also have a little brother named Leo who's amazing as well. I'll just give, as I indicated, a brief overview of from a historical perspective. I worked with Julie Axelrod on small molecules, neurotransmitters, and in those days, based on the work of Julie and numerous other people, we knew a great deal about how neurotransmitters are made, how they're degraded, how they're inactivated by reuptake into the nerve ending that released them, but there was very little, if anything, done about the actual receptor site where they worked. And I got involved in that quest because President Nixon declared war on heroin in 1971 and appointed a fellow named Jerry Jaffe as the czar of all heroin, all drug abuse in America. And I didn't know morphine from marijuana, but I knew Jerry Jaffe, and successfully convinced him to set up drug abuse research centers. And even though I'd never worked on drug abuse, we got one of the centers and figured out we had to do something about it. And this notion that there had to be a receptor for opiates came along, and that led to efforts to identify opiate receptors. What this slide indicates is how similar opiate agonists like morphine are to antagonists like nalorphine, differing only in an N-methyl group substituent, and yet these very, very different properties, which turned out, once it was possible to measure opiate receptors, depended on subtleties of interactions with the receptor complex involving G proteins, sodium, and things else like that. The direct evidence for opiate receptors being pharmacologically relevant and associated with the binding sites came from simple-minded strategies like this one, in which the relative pharmacologic potency of opiates indicated in this ability to inhibit electrically induced contractions of the guinea pig intestine paralleled their affinity for the binding sites, and pretty compelling. And we thought we were dealing with real live opiate receptors. And then, of course, the question came that man was not born with morphine in him. Why do we have opiate receptors? Is there an endogenous ligand? And several labs throughout the world looked for what could be the endogenous ligand. We looked for compounds in the brain, extracts that would inhibit binding of radioactive opiates to opiate receptors and whose relative densities throughout the brain paralleled the density of opiate receptors. In Aberdeen, Scotland, Hans Kosterlitz and John Hughes, who were smooth muscle pharmacologists, looked for brain extracts to mimic the ability of morphine to inhibit electrically induced contractions of the mouse vas deferens, and they won the race and identified the enkephalins, which I won't show you here. Oh my God, I apologize. That's the opiate receptors. But here is the evidence that enkephalins, as isolated by Kosterlitz and Hughes, are indeed the endogenous ligands. And this is one line of evidence, namely the extraordinarily close coincidence of the localization of enkephalins and opiate receptors. And that and lots of other evidence convince us and others that those are the endogenous ligands for opiate receptors, and that simple-minded receptor binding technology could be applied to lots of other receptors. For instance, one effort was to try and find out how dopamine acts. Another question was, of course, how do antipsychotic drugs act? And this is a depiction of chlorpromazine, the parent of all antipsychotic neuroleptic drugs, and Alan Horn in our lab showed the similarity in the optimal conformation of chlorpromazine and that of dopamine. And it wasn't long thereafter before Ian Crease in our lab identified the dopamine receptors by simple-minded ligand binding, and utilizing this simple-minded correlational study provided pretty compelling evidence that this is the site at which the antipsychotic drugs do their main thing. And as Rusty indicated, studies of neurotransmitter receptors by ligand binding techniques were going on in the mid-1980s when Michael Berridge made the remarkable discovery that besides cyclic AMP, there was another major second messenger system, inositol trisphosphate, inositol 1,4,5 trisphosphate, or IP3, acts intracellularly to release calcium, and molecular identification of exactly what was going on and how it worked wasn't very evident, but we used a simple-minded ligand binding to find receptors for IP3. IP3 is indicated here on this slide, inositol 1,4,5 trisphosphate. What this slide indicates, though, is that there's a whole slew of other inositol phosphates, higher inositol phosphates, every one of which has significant biological importance. What's highlighted here, especially, is IP7, because IP7 and IP8 are notable in that you have a diphosphate bond, which has that little mark indicating energy. In fact, it's just as energetic as ATP. We obtained some evidence that IP7 can phosphorylate proteins. Whether it does it in real life, we don't know, but clearly there's the whole world of insight into the inositol phosphates, which do a lot of different things. Indicated here, one of the enzymes, IPMK, besides being the physiologic enzyme that generates IP5, Adam Resnick in our lab and David Mag as well discovered that it's a PI3 kinase, which works in tandem with the P110 PI3 kinase, characterized elegantly by Lewis Cantley. And that's the tip of the iceberg. IPMK, non-catalytically, is a transcriptional coactivator for a variety of targets. The study of small molecules led us into investigations of gases as neurotransmitters, as was indicated already. And this slide depicts a number of interesting concepts. One shows how neurotransmission by glutamate acting at NMDA receptors can signal via activating nitric oxide synthase that David Brett, an MD PhD student in our lab, discovered. Calcium depolarize the cell by glutamate acting at NMDA receptors or by other stimuli, and calcium enters the cell, binds to calmodulin, and calmodulin calcium activates the formation of nitric oxide from the amino acid arginine. And then one of the targets is stimulation of soluble guanyl cyclase. At the bottom of the slide is indicated a parallel pathway in which carbon monoxide can also be a neurotransmitter, work by A.J. Verma, an MD PhD student in our lab. And here it's very interesting. It comes from the enzyme from heme, which is in aging red blood cells, heme has to be degraded, and it's done so by an important enzyme called heme oxygenase, which is inducible, or it's heme oxygenase number 1, or HO1. And that breaks open the heme ring, and you get biliverdin and then bilirubin. In neuronal systems, there's a different enzyme which is also abundant. It's called heme oxygenase 2, or HO2. And here it's depicted generating carbon monoxide. Recently, work in our lab has focused on the other products. We already knew that bilirubin, it's an antioxidant, and David Bargnano in our lab established it pretty much as a physiologic cytoprotectant. Very recently, in work not yet published, Majid Haraz in our lab has shown bilirubin is acting and regulating autophagy at picomolar concentrations. So it's a completely different story from the conventional actions of bilirubin that are actually generally at millimolar ranges. And very, very recently, in work not yet published, Majid Haraz and Jimmy Mejan addressed the question of itching. And why do they care about itching? Well, they care about itching because Xingshuang Zhang, who was a faculty member in our department, in fact, the last faculty member I recruited before stepping down as chair of the department, and one of his claims to fame is identifying the family of MERG receptors that he started out as a postdoc with David Anderson identifying, and these are itch receptors. And Jimmy Mejean was working on them, and Shirek Vasavta in our lab said, you know, bilirubin that we study in our lab might have something to do with itching. If you have jaundice, you have elevated bilirubin, and you itch a lot. And nobody ever established, well, why do you itch when you have jaundice? And Girard said, well, the simple-minded possibility, based on everything we know, is maybe bilirubin makes you itch, but nobody had ever looked at that. And so, very recently, Chirag and Jimmy looked at it, and you put in bilirubin in the skin, and it makes animals itch. And so, since Jimmy was studying the itch receptor based on the work of his mentor Xingshuang Zhang, and here's a description of their paper elucidating the itch receptor, he said, well, let's see what happens. They already had, based on Xingshuang Zhang's work, we had mice with knockout of a whole family of the MERG receptors. And to make a long story short, you knock out the MERG receptors, and then bilirubin, well, in this case, itching in general, is eliminated. Okay. Now, so as you can see, one of the themes of the work in our lab has always been to do basic molecular neuroscience, but always with a view to disease relevance. And one of the diseases that's intrigued us in recent years is Huntington's disease, which is summarized here. As you know, it's a autosomal dominant disease with totally fatal, caused by this triplicate repeat that's very well known. Polyglutamine, if you have more polyglutamines, you get a worse disease and earlier onset. And one of the big questions about Huntington's disease had always been, why is it that, as you see in this picture here, you get massive and almost total loss of the caudate nucleus that regulates motor behavior. That's why people with Huntington's disease have Huntington's chorea. And why is there, as the disease advances, the whole brain deteriorates, but for quite a while there's just very selective destruction of the corpus striatum, and nobody knew why. So Srini Subramanian, a postdoc in our lab, asks the question of, well, could there be something that's uniquely concentrated in the corpus striatum that might interface with Huntington? And he had noticed literature on a small G protein called RACE, which didn't have its, in fact, the name RHES, RACE, stands for RAS homolog enriched in the striatum. And to make a long story short, he did a very elegant body of work, several lines of evidence indicating that RACE explains the selective destruction of the corpus striatum in Huntington's disease due to its interactions. And the proof of the pudding is studies by Biotin et al., as well as Robbie Mueller, an MD PhD student in our lab, establishing that you knock out RACE and the Huntington's disease, you make it better in these various. In one case, it's a genetic model of Huntington's disease. In the other case, it's a chemically induced model of the disease. So now we're talking about Huntington's disease. Let's talk about another neurotransmitter system that seems to have some relevance. And this has to do with hydrogen sulfide that we've been looking at recently. And this is based on work that we initiated in collaboration with Rui Wang in Canada some years ago, in which we found that hydrogen sulfide is physiologically generated. Hydrogen sulfide had been detected in biological tissues for years, but nobody ever knew whether it normally occurs biologically or just there from exogenous sources. And there's a couple of enzymes that in theory could make it. One is called cystathionine beta synthase that makes the weird dipeptide cystathionine, and the other is called cystathionine gamma lyase or cystathionase that destroys it, and that destroys cystathionine. And we focused on cystathionase or cystathionine gamma lyase, which we abbreviate as CSE for cystathionase. And to make a long story short, cystathionase, this particular paper that was in Science in 2008, we obtained evidence that hydrogen sulfide has endothelial-derived relaxing factor activity, which in some cases look to be just as equal as important as nitric oxide's activity. Other cases maybe not. But this is the title of the paper, and you can see the evidence that it acts like an EDRF, and the enzyme is localized to the endothelial layer of the blood vessels. And the important thing is that it makes H2S, and that we have knockout mice that can be employed to characterize it. So that's how hydrogen sulfide is made. How does hydrogen sulfide signal? And again, we used analogies with the nitric oxide system. Nitric oxide, I already showed you, can relax blood vessels by activating soluble guanylyl cyclase in blood vessels. But Jonathan Stamler then did a nice body of work indicating that it could also nitrosylate cysteines in proteins. It's just, the NO would react with the SH of the cysteine. You have an NO-S complex, usually inactivating things. And it wasn't clear whether that happened in real life because he couldn't measure it in regular biological tissues. Sami Jaffray in our lab developed what's called the Biotin switch assay, where you could routinely measure nitrosylation, and found that there is indeed physiological nitrosylation that can be activated by neurotransmitter systems. And I won't go into the details about that at this time because we're going to focus on hydrogen sulfide. So analogous to nitrosylation, which is depicted here, is the possibility of sulfhydration, like an analogous, similar sort of thing. And Asif Mustafa, another MD PhD student in our lab, showed that sulfhydration indeed takes place. It's reversible by DTT. In this case, it's elicited by NAHS. But on the right-hand side of your slide, you can see what's important in terms of physiologic activity. Namely, if you take glyceraldehyde 3-phosphate dehydrogenase, the major glycolytic enzyme GAPDH, and measure its catalytic activity, and you treat it with hydrogen sulfide, you triple catalytic activity, and that's also reversed by DTT. What's important is whether that happens in real life. So how would we study whether H2S is regulating GAPDH and other enzymes in real life? Well, we have our mice with knockout of CSC, the enzyme that makes H2S. And I don't have the slide here to show you, but if in those mice the catalytic activity of GAPDH is reduced by about 35–40%, even though the amount of GAPDH protein hasn't changed. In other words, that's direct evidence that it's the H2S sulfhydrating the GAPDH that maintains its basal activity to a pretty darn major extent. And in other work that I don't have time to show you today, it turns out that the majority of proteins that you study are physiologically sulfhydrated to a pretty significant extent, substantially more than nitrosylation. Sulfhydration is a bigger signaling system than nitrosylation. Here's just a few examples from our lab of processes. So the anti-apoptotic actions of NF-kappa B are mediated by hydrogen sulfide sulfhydration, and ditto, the neuroprotective actions of Parkin can be attributed to that. Now let's get back to Huntington's disease that I was telling you about. This is work primarily of Bindu Paul and Juan Sabodio, postdocs in our lab. What happened was that Bindu was studying CSE, this enzyme that makes hydrogen sulfide, and cysteine, by the way, because cysteine is physiologically generated from cystathionine by CSE, which generates H2S from cysteine as well. She was looking at the CSE knockout mice and just holding them up, and they were strange. Other people in the lab were working with mouse models of Huntington's disease. And she was looking at those mice, and she said, they look remarkably similar. It looks as if, so as she speculated, maybe CSE deficiency is what gives this weird phenotype, and maybe that might be abnormal in Huntington's disease. And as you can see here, in Huntington's disease, the number of glutamine repeats you have is key. 7 glutamine repeats are completely normal. 111 is you got a robust form of Huntington's disease, and in cell lines, the Q7 and Q111 cell lines are the routine forms studied for Huntington's disease. And if you look at the Q111 cell lines, you can see there's a massive depletion of CSE, and that's for Q111 Huntington's disease. More important, if you look at the bottom of the slide, for patients with Huntington's disease, CSE protein is massively depleted. And it's not just because you have brain rot in Huntington's disease, because beta-actin is quite normal. So, what regulates CSC? And that's what we've been working on very recently to a significant extent, largely through the efforts of Juan Sabodio and Bindu Paul. Well, CSE is regulated by a couple of transcription factors. SP1 is one of them. That's depicted here. And so could it be the case that the CSE deficiency has something to do with transcription? Well, CSE messenger RNA is also affected in Huntington's disease. And SP1 is disrupted in Huntington's disease as well. So that fits with the model that perhaps mutant Huntington would sequester SP1 to prevent transcription of CSE. And the proof is in the pudding. As you can see here, I didn't show you, I left out the slide, but basically the SP1 stuff is altered significantly. And now I'm going to finish up by just focusing on another transcription factor for CSE, which is especially regulating CSE under stress. It's called ATF4. And you can see that ATF4 is depleted in a major way with the Q111 Huntington's disease mutation in parallel with the depletion of CSE itself. This is all linked to the condition of low cysteine. So cysteine, if you have low cysteine, you induce ATF4. with low cysteine normally. But if you have Huntington's disease, low cysteine doesn't do nothing. On the other hand, that's with other amino acids. In other words, here again, low cysteine induces ATF4. Low arginine, lysine, glutamine, leucine also induce ATF4. In Huntington's disease, arginine, lysine, glutamine, leucine deficiencies also induce ATF4, but cysteine doesn't do it. So it's very specific. This abnormality is very notable and very notably selective for the cysteine, low cysteine conditions. We still don't know exactly what the mechanism is that's accounting for the abnormality in Huntington's disease with ATF4. However, we have a hint. We know that there's a lot of oxidative stress in Huntington's disease. So the question is, would oxidative stress account for the low inducibility of ATF4? And the answer is that's indeed the case. Oxidative stress turns down the ATF4 induction by low cysteine. It does not affect induction by thapsigargin, which I don't show you here. But the low cysteine, the loss of inducibility by low cysteine is by oxidative stress is reversed by antioxidants. So the question is, does any of this matter? So we've established that there is this abnormality in CSE, in Huntington's disease, and we've talked about the molecular mechanisms involving the transcription factor ATF4 and SP1. What would happen if you said, well, I mean, how could we test the hypothesis that this is responsible for the symptoms of Huntington's disease? Well, I suppose we could replace the missing cysteine. So here is again our Huntington's disease Q111 cells, and you can see again the depletion of cysteine, the depletion of its catalytic ability to make hydrogen sulfide, the depletion of its ability to generate cysteine from cystathionine. Then, if we put back cysteine, all of those things get better. And more importantly, the proof is in the pudding. Clinically, in this study of Bindu's that was in Nature last year, you can see, just look at those ventricles. You can estimate brain degeneration by the increased ventricular size. And you can see in R62, Huntington's disease, there's degeneration, big enlarged ventricles. If you treat with N-acetylcysteine, you actually can reverse the neuropathology and you can prolong the life of these animals. And so actually this work is now leading to clinical trials in Huntington's disease of N-acetylcysteine, which seems to be interesting. So as I think you can see, neurotransmitters, especially the gazotransmitters, do good things and may even have clinical relevance. And it's all due to these wonderful people in the lab. Here is Bindu Paul, who is the mastermind behind a lot of the Huntington's disease work, together with her partner, Juan Sabodio. Here are other people. Here is Srini Subramanian, who did the work showing that race accounts for the reason that Huntington's disease destroys the corpus striatum selectively. And this is a family that's been together for a while. So Adele Snowman graduated the University of Connecticut in June of 1972, came to work with me in September of 1972, and was a dynamo who would come in at 5:30 in the morning and stay till 8 o'clock at night. And she arrived in the lab the same week that a graduate student, Candace Pert, had identified binding of opiates to what was the opiate receptor. And it was clear there was a lot of stuff to be done. And by having Adele, who worked like 5 people, better than 5 people together, that work got turned on. Linda Hester came in 1977 and continues to run everything that we use, cell culture, has been her work. Here's Asif Mustafa, who did a lot of the hydrogen sulfide work that I alluded to. And there's a bunch of other terrific people who've done different projects in the lab. And they're what makes life worth living and makes it fun to come to the lab every day. And I thank you.
We have time for some questions. Linder.
So, in the trial with Huntington, with the NAC, do you go for people like in Colombia or so where they know these patients will get Huntington, so that the effect is much greater versus those who have Huntington?
You're asking about the trial of N-acetylcysteine?
Right.
Yeah, it's a good question. We're in discussions with various clinicians to see what's the best kind of population to do that kind of clinical trial.
When did you start treating the animals to get the reversal? Was it a reversal or protection?
It was mostly protection, and I can't remember the exact time.
But it was early.
Yeah.
Early on. Mm-hmm.
Yeah.
Yeah, Tony.
So, Saul, do you think there are any additional neurotransmitters yet to be discovered?
Whenever anybody says we've got too many neurotransmitters, then you know we don't have enough. I remember when, in fact, Vernon Mountcastle, one of the fathers of modern neuroscience, was a colleague and tennis partner at Hopkins, and he liked to poo-poo us biochemical types, saying, well, you know, as a neurophysiologist, we need one excitatory and one inhibitory neurotransmitter, so all these other silly chemicals you're looking at are just fluff. But they're, so in general, I'd prefer to think we've only scratched the tip of the iceberg.
And I guess related to that would be, you know, the surprises of the endogenous molecules like the opiates and the cannabinoids.
Yeah.
Are there any others on the horizon that are going to surprise us?
Nothing that I can think of to talk about. Yeah.
Yeah, Joe.
Related to that, so there's been, I guess it's somewhat controversial that mice, at least mice, maybe humans, have the capacity to make their own morphine, the actual molecule.
They're on morphine.
Yeah, I've heard saying it's over the years, showed some evidence this is the case. Where do you come down on this?
Yeah, so what the question refers to is there have been publications going back to the late 70s even, I forget exactly when, people saying they had isolated morphine itself from mammalian brain or other tissues. And there's a problem. First problem is that if you look at the structure of morphine, you'd say, there aren't, I don't know all the enzymes in the body, but it's hard to conceptualize of any enzyme that could do what is necessary to do to create morphine. And then there were papers saying that the isolation of so-called endogenous morphine was due to morphine, that there's morphine in bottles in the lab and all that. And when you take, 100 kilograms of brain, you'll end up with a little bit of morphine. And so we've never done any work in our lab on that, but I gather it's controversial and just based on common sense reasoning, I don't think it's that likely.
Among the trainees that Saul's had in his lab was Peter Salk, is that right? Was he,
he was an advisee. He didn't ever work in our lab.
Is that right?
Yeah.
You were telling me earlier about one of the first times you visited Salk in the 60s.
Yeah.
Do you remember anything about who was there at the time? 68, was it?
Yeah, roughly 68. And the anecdote I was relating to earlier today was that at that time we had done molecular modeling to try and understand why some psychedelic drugs like mescaline did the same thing as LSD, though chemically mescaline looked very different than LSD, at least superficially. And we'd done some molecular modeling to show, well, maybe they really did resemble each other. And there was a meeting at the Salk Institute. I was just a very young assistant professor and gave this presentation about how structurally with writing models, the mescaline, LSD, and the other psychedelic drugs could look like each other. And the great chemist Leslie Orgel raised his hand and he said, well, you know, according to conventional chemistry about what's an optimal conformation, everything you have is horseshit. Being British, he used more elegant words. And he gave me, you know, umpteen reasons why, and of course I'm not a chemist, so I just sort of mumbled a little bit, and as I was trying to mumble and try and figure out an answer, a big tall guy in the audience stands up named Francis Crick, and he said, Leslie Orgel, you have no creativity. You're talking about very conventional chemical conformations of molecules, and what Dr. Snyder is talking about is something that's unconventional, and it's the unconventional things that really matter and are for real, and that's where the future is, and why don't you shut up? So I had a warrior on my side, and Francis Crick, and we became good friends thereafter.
All right, thanks very much, Saul.